So in your body, you have a few different overarching types of cells. You've got, for one, you've got your cells that are actively able to divide. So some examples would be the epithelial cells that make up your skin, and you've got your fibroblast cells that make up the scaffolding of most of your organs, like say your kidneys and your liver, and even the endothelial cells that line the insides of your blood vessels. So these cells, right, and their precursors, they can all divide by mitosis, and that's to help replenish and regenerate the tissues that they're a part of, right? And actually, we'll include some of our stem cells in here too, the cells that these other cells initially come from. And we'll call this whole set of cells here mitotic cells. They undergo mitosis. So we also have another group of cells in our bodies that we refer to as non-mitotic cells, or actually post-mitotic cells. That's a more accepted name. And you can probably guess why. They don't undergo mitosis. They're incapable of proliferation. And you might sensibly be thinking that, well, hey, that means that they can't repair and regenerate like our mitotic cells over here. And you'd be on the right track. They certainly have a limited ability to repair or regenerate the tissues that they're a part of. And some examples would be, say, neurons, either within the brain or elsewhere in your nervous system, and heart muscle cells, right? These are neurons and heart muscle cells. These are both examples of post-mitotic cells. But yeah, you're on the right track. This tissue doesn't regenerate by mitosis of these cells, like what happens over on this side here. This tissue over here regenerates only very slowly, and only by asking some special tissue-specific stem cells to help them out. But it's a slow process of repair in post-mitotic tissues, so to speak. So here's the thing, though. Here's the problem with mitotic division. So what the cells on this side do. So with mitosis comes DNA replication, because in order to create new daughter cells, you have to copy your DNA to pass on, right? But DNA replication in basically every mitotic cell, besides our embryonic stem cells, has this interesting little caveat. So let me show you what I mean here. So here's our cell ready to start mitosis, right? So it wants to copy its DNA to pass a full set on to each of its daughter cells. So here's our DNA. Remember, in eukaryotes like us humans, we have nice linear strands of DNA, and we have lots of them, as opposed to our prokaryotic bacteria friends who have circular DNA. So here's our linear DNA, and actually at the ends of our DNA strands, right, on both sides, we have these little caps, sort of like the little plastic tips on the ends of your shoelaces. And just like how the little plastic tips protect your shoelaces from fraying or getting worn out, our little telomeres, that's what these tips are called, our telomeres prevent damage from happening to our DNA during the copying process. So to clarify, these telomeres at the ends here don't actually code for any proteins. They don't have any genes on them. All the important DNA is in this section here, this middle section. So I said that there's potential DNA damage during the copying process. What do I mean by that? Well, the machinery that copies our DNA polymerase system, it kind of works in a funny way. It doesn't actually copy our DNA all the way to the end. So what we see is that each time DNA is copied, you lose a little bit of telomere here. So every time the DNA gets copied, you lose a little bit more and a little bit more and a little bit more of the end. So lucky for us, instead of having really important coding DNA at the end of our chromosomes, we've got our telomeres. So again, it's the telomeres that end up becoming slightly shorter at the end of each DNA replication cycle. And it turns out that a cell can undergo about 60 or 70 or so cell divisions before the telomeres get too short, and then all this important coding DNA here starts to become at risk of damage from further replications. But not to worry, we have a contingency plan built in. So once the telomeres get too short, the cell initiates a DNA damage response. So it sort of rings these internal alarm bells, and it sort of gives up its ability to divide, because it really wants to prevent any more shortening of this chromosome that would happen in further rounds of replication, right? So it loses its ability to divide. And by doing that, it has become what's called a senescent cell. And before I explain what exactly that is, there's just two things I want to point out about this. So the first thing is that if a cell reaches senescence because its telomeres have become too short, we say it's reached replicative senescence. And the second thing is that the number of times a cell can divide before reaching senescence, replicative senescence, is called its Hayflick limit. And that's named after the scientist who figured that out. And again, it's generally around about 60 or so divisions. So now that we've gotten all that out of the way, what exactly is senescence? Well, basically it's a change in a cell's state from a happy, active, mitotic cell, right, i.e. one that divides on a regular basis, to one that's non-dividing and possibly less happy. I'll have to ask a senescent cell how it's feeling one of these days. But it undergoes all these other changes as well. Like it starts expressing all sorts of genes that it didn't before, and it starts to look a little bit different, and it starts to respond a bit differently to the cells around it. But the important thing is that while it's still contributing to the structure of your body, it's still hanging around in your tissues, it's not allowed to replicate anymore. And kind of the key with all roads to cellular senescence, be it through reaching your Hayflick limit, as we just saw, or through other ways, which I'll mention in a second, the key is that cells transform into a senescent state to prevent impending DNA damage from happening and causing maybe cancer or a tumor, or possibly being passed on to daughter cells. So the cell's ability to divide gets turned off. And other causes appear. Well, essentially anything that threatens the integrity of your DNA. So maybe if you had telomeres that just don't work properly, or maybe you sustained some DNA damage through a mutation of some type, or maybe some toxin damaged your DNA, any of these could really just as easily result in your cell becoming senescent, non-dividing. So is there actually a problem with senescent cells? Are we happy about them? Are we upset with them? What's the deal here? Well, we're not really sure yet, but conceptually an easy way I like to think about them is that, well, on one hand, they help prevent tumors or cancers from happening because they're not allowed to divide anymore. But on the other hand, as we get older and our tissues have more and more senescent cells in them, the tissue can't really repair itself as well as it used to because there's fewer actively mitosing cells around, right? And there's actually also some evidence linking senescent cells to age-related diseases like cataracts. But this is a pretty active area of research. So for now, we'll just have to keep an eye on these cells to see what new research comes up with for us. Now, let me zoom out a bit here to show you something else. So here's our mitotic cells, right, that by definition are the only ones that can undergo replicative senescence. And over here are our post-mitotic cells. And these guys can't reach replicative senescence, right, because they don't replicate, but they can be induced to become senescent if their DNA is at risk of becoming damaged in basically just the same ways I mentioned earlier on, DNA damage from some source. And the last thing, I just want to draw out a graph here to represent our different types of cells and their tendencies with division. I think this will really help us to get the picture here. So here's our graph. Here's our y-axis here, and here's our x-axis. And on the y-axis, I want to put cell division capacity, okay, so capacity of this cell to divide. And on the x-axis here, I'll put number of doublings, which is essentially how many times the cell has divided already. And this will all sort of make sense in a second here. So our somatic cells, our regular body cells that are able to mitose, to undergo mitosis, they start out, say up here, right, with a pretty high cell division capacity. They're able to divide a lot. And as they continue to divide and divide and divide, they sort of move this way. Their curve sort of curves down this way as the number of doublings that they've done increases, right? So you can see the more doublings they've done, the lesser their cell division capacity, right? And that's because their telomeres, with each doubling, their telomeres are getting shorter. So their capacity to divide is getting lower with the increased number of doublings, okay? But we have another type of cell, right? We have, say, some of our stem cells. They start off up here, right? Let's say they start off at the same place. They have a really high cell division capacity. But as they divide, right, their line, their curve just stays really high, right? Their cell division capacity, regardless of the number of doublings, regardless of the number of times they've divided, their cell division capacity stays really high, right? So why would that be? Why might that be? Why would they be so different from our somatic cells? Well, it turns out that they express a special enzyme called telomerase. And what telomerase does is every time one of these cells from our stem cell curve here every time it undergoes replication and loses a bit of telomere, telomerase, the enzyme, sort of springs into action and adds back that little bit of telomere, right? So that means that these cells are never going to reach a Hayflick limit, right? They're just going to have the ability to replicate as many times as they want. And that's because they express telomerase. And the very last thing I'll say is that sometimes your somatic cells, right, they can develop a mutation in their DNA that causes them to start expressing telomerase. So now they have the enzyme telomerase present within the cell, right? So now they jump off our curve here, our regular somatic cell curve here, because they get the ability to replicate as many times as they want. They essentially escape senescence, and this is one of the ways that cancer occurs, because now you have a group of cells that can go on and multiply out of control and form a tumor.
Telomerase function. The ends of chromosomes contain regions referred to as telomeres. These telomeric regions consist of telomeric repeat sequences. The exact sequence of telomeric repeat can vary from species to species. The repeat sequence shown here is found on telomeres of human chromosomes. Since DNA replication can only proceed in the 5' to 3' direction and requires a primer, it is not possible for a DNA polymerase to replicate the 3' ends of linear chromosomes. If this problem were not corrected, chromosomes would shorten with each cycle of DNA replication. The addition of telomeric repeat sequences by an enzyme complex referred to as telomerase assures that this shortening does not occur. Telomerase is a unique and interesting enzyme because, in addition to having DNA polymerase activity, it also contains an RNA sequence that provides a template for the synthesis of telomeric repeat DNA. Part of the RNA sequence hybridizes with the single-stranded overhang on the DNA strand, leaving a single-stranded overhanging RNA sequence. The repeat unit shown here is from Tetrahymena, which is the model organism used in telomere and telomerase research. The DNA polymerase function of telomerase then synthesizes a DNA strand complementary to the RNA found in the telomerase and subsequently translocates to the end of the newly synthesized strand, and the process repeats itself. Many cycles of repeats can occur. Once telomerase has completed its function, DNA primase synthesizes an RNA primer near the 3' end, and DNA polymerase fills in the vacant region. A short region at the 3' end will remain single-stranded. The end result is that telomerase will have added many repeat sequences from a few dozen to a few hundred. This prevents chromosome shortening.
Where does the end begin? Well, for me, it all began with this little fellow. This adorable organism, well, I think it's adorable, is called Tetrahymena, and it's a single-celled creature. It's also been known as pond scum. So that's right, my career started with pond scum. Now, it was no surprise I became a scientist. Growing up far away from here, as a little girl, I was deadly curious about everything alive. I used to pick up lethally poisonous stinging jellyfish and sing to them. And so, starting my career, I was deadly curious about fundamental mysteries of the most basic building blocks of life. And I was fortunate to live in a society where that curiosity was valued. Now, for me, this little pond scum critter, Tetrahymena, was a great way to study the fundamental mystery. I was most curious about those bundles of DNA in our cells called chromosomes. And it was because I was curious about the very ends of chromosomes, known as telomeres. Now, when I started my quest, all we knew was that they help protect the ends of chromosomes. It was important when cells divide. It was really important. But I wanted to find out what telomeres consisted of. And for that, I needed a lot of them. Now, it so happens that cute little Tetrahymena has a lot of short linear chromosomes, around 20,000. So lots of telomeres. And I discovered that telomeres consisted of special segments of non-coding DNA right at the very ends of chromosomes. But here's a problem. Now, we all start life as a single cell. It multiplies to 2, 2 becomes 4, 4 becomes 8, and on to form the 200 million billion cells that make up our adult body. And some of those cells have to divide thousands of times. In fact, even as I stand here before you, all throughout my body, cells are furiously replenishing to, well, keep me standing here before you. So every time a cell divides, all of its DNA has to be copied, all of the coding DNA inside of those chromosomes, because that carries the vital operating instructions that keep our cells in good working order. So my heart cells can keep a steady beat, which I assure you they're not doing right now, and my immune cells can fight off bacteria and viruses, and our brain cells can save the memory of our first kiss and keep on learning throughout life. But there is a glitch in the way DNA is copied. It is just one of those facts of life. Every time the cell divides and the DNA is copied, some of that DNA from the ends gets worn down and shortened, some of that telomere DNA. And think about it, like the protective caps at the ends of your shoelace, and those keep the shoelace or the chromosome from fraying. And when that tip gets too short, it falls off, and that worn down telomere sends a signal to the cells. The DNA is no longer being protected, sends a signal, time to die. So, end of story. Well, sorry, not so fast. Can't be the end of the story, because life hasn't died off the face of the earth. So I was curious, if such wear and tear is inevitable, how on earth does Mother Nature make sure we can keep our chromosomes intact? Now remember that little pond scum critter, Tetrahymena? The craziest thing was Tetrahymena cells never got old and died. Their telomeres weren't shortening as time marched on. Sometimes they even got longer. Something else was at work, and believe me, that something was not in any textbook. So, working in my lab with my extraordinary student, Carol Greider, and Carol and I shared the Nobel Prize for this work, we began running experiments, and we discovered that cells do have something else. It was a previously undreamed of enzyme that could replenish, make longer telomeres. And we named it telomerase. And when we removed our pond scum's telomerase, their telomeres ran down and they died. So it was thanks to their plentiful telomerase that our pond scum critters never got old. Okay, now that's an incredibly hopeful message for us humans to be receiving from pond scum, because it turns out that as we humans age, our telomeres do shorten, and remarkably, that shortening is aging us. Generally speaking, the longer your telomeres, the better off you are. It's the overshortening of telomeres that leads us to feel and see signs of aging. My skin cells start to die, and I start to see fine lines, wrinkles. Hair pigment cells die. You start to see gray. Immune system cells die. You increase your risks of getting sick. In fact, the cumulative research from the last 20 years has made clear that telomere attrition is contributing to our risks of getting cardiovascular diseases, Alzheimer's, some cancers, diabetes, the very conditions many of us die of. And so we have to think about this. What is going on? This attrition, we look and we feel older. Yeah, our telomeres are losing the war of attrition faster. In those of us who feel youthful longer, it turns out our telomeres are staying longer for longer periods of time, extending our feelings of youthfulness and reducing the risks of all we most dread as the birthdays go by. Okay, seems like a no-brainer. Now, if my telomeres are connected to how quickly I'm going to feel and get old, if my telomeres can be renewed by my telomerase, then all I have to do to reverse the signs and symptoms of aging is figure out where to buy that Costco-sized bottle of grade A, all-organic, fair trade telomerase, right? Great, problem solved. Not so fast. I'm sorry. Alas, that's not the case. Okay. And why? It's because human genetics has taught us that when it comes to our telomerase, we humans live on a knife edge. Okay, simply put, yes, nudging up telomerase does decrease the risks of some diseases, but it also increases the risks of certain and rather nasty cancers. So even if you could buy that Costco-sized bottle of telomerase, and there are many websites with marketing such dubious products. Problem is, you could nudge up your risks of cancers. And we don't want that. Now, don't worry. And because, well, I think it's kind of funny that right now, many of us may be thinking, well, I'd rather be like pond scum. Right. There is something for us humans in the story of telomeres and their maintenance. But I want to get one thing clear. It isn't about enormously extending human lifespan or immortality. It's about health span. Now, health span is the number of years of your life when you're free of disease, you're healthy, you're productive, you're zestfully enjoying life. Disease span, the opposite of health span, is the time of your life spent feeling old and sick and dying. So the real question becomes, okay, if I can't guzzle telomerase, do I have control over my telomeres length and hence my well-being, my health, without those downsides of cancer risks? Okay? So it's the year 2000. Now, I've been minutely scrutinizing little teeny tiny telomeres very happily for many years when into my lab walks a psychologist named Elissa Epel. Now, Elissa's expertise is in the effects of severe chronic psychological stress on our minds and our body's health. And there she was standing in my lab, which ironically overlooked the entrance to a mortuary, and and she had a life and death question for me. What happens to telomeres in people who are chronically stressed? She asked me. You see, she'd been studying caregivers, and specifically mothers of children with a chronic condition, be it gut disorder, be it autism, you name it, a group obviously under enormous and prolonged psychological stress. I have to say, her question changed me profoundly. See, all this time I'd been thinking of telomeres as those minuscule molecular structures that they are and the genes that control telomeres. And when Elissa asked me about studying caregivers, I suddenly saw telomeres in a whole new light. I saw beyond the genes and the chromosomes into the lives of the real people we were studying. And I'm a mom myself. And at that moment, I was struck by the image of these women dealing with their child with a condition very difficult to deal with, often without help. And such women simply often look worn down. So was it possible their telomeres were worn down as well? So our collective curiosity went into overdrive. Elissa selected for our first study a group of such caregiving mothers, and we wanted to ask, what's the length of their telomeres compared with the number of years that they have been caregiving for their child with a chronic condition? So 4 years go by, and the day comes when all the results are in. And Elissa looked down at our first scatter plot and literally gasped, because there was a pattern to the data. And it was the exact gradient that we most feared might exist. It was right there on the page. The longer, the more years, that is, a mother had been in this caregiving situation, no matter her age, the shorter were her telomeres. And the more she perceived her situation as being more stressful, the lower was her telomerase and the shorter were her telomeres. So we had discovered something unheard of. The more chronic stress you're under, the shorter your telomeres, meaning the more likely you were to fall victim to an early disease span and perhaps untimely death. Our findings meant that people's life events and the way we respond to these events can change how you maintain your telomeres. So telomere length wasn't just a matter of age counted in years. Alyssa's question to me back when she first came to my lab indeed had been a life and death question. Now, luckily, hidden in that data, there was hope. We noticed that some mothers, despite having been caring for their children for many years, had been able to maintain their telomeres. So studying these women closely revealed that they were resilient to stress. Somehow they were able to experience their circumstances not as a threat day in and day out, but as a challenge. And this has led to a very important insight for all of us. We have control over the way we age, all the way down into our cells. Okay, now our initial curiosity became infectious. Thousands of scientists from different fields added their expertise to telomere research, and the findings have poured in. It's up to over 10,000 scientific papers and counting. So several studies rapidly confirmed our initial finding that, yes, chronic stress is bad for telomeres. And now many are revealing that we have more control over this particular aging process than any of us could ever have imagined. A few examples. A study from the University of California, Los Angeles, of people who are caring for a relative with dementia long-term, and looked at their caregivers' telomere maintenance capacity and found that it was improved by them practicing a form of meditation for as little as 12 minutes a day for 2 months. Attitude matters. If you're habitually a negative thinker, you typically see a stressful situation with a threat-stress response, meaning if your boss wants to see you, you automatically think, I'm about to be fired, and your blood vessels constrict, and your level of the stress hormone, cortisol, creeps up, and then it stays up. And over time, that persistently high level of the cortisol actually damps down your telomerase, not good for your telomeres. On the other hand, if you typically see something stressful as a challenge to be tackled, then blood flows to your heart and to your brain, and you experience a brief but energizing spike of cortisol. And thanks to that habitual bring-it-on attitude, your telomeres do just fine. So, what is all of this telling us? Your telomeres do just fine. You really do have power to change what is happening to your own telomeres. But our curiosity just got more and more intense because we started to wonder, what about factors outside our own skin? Could they impact our telomere maintenance as well? You know, we humans are intensely social beings. Was it even possible that our telomeres were social as well? And the results have been startling. As early as childhood, emotional neglect, exposure to violence, bullying, and racism all impact telomeres, and the effects are long-term. Can you imagine the impact on children of living years in a war zone? People who can't trust their neighbors and who don't feel safe in their neighborhoods consistently have shorter telomeres. So your home address matters for telomeres as well. On the flip side, tight-knit communities, being in a marriage long-term, and lifelong friendships even, all improve telomere maintenance. So what is all this telling us? It's telling us that I have the power to impact my own telomeres, and I also have the power to impact yours. Telomere science has told us just how interconnected we all are. But I'm still curious. I do wonder what legacy all of us will leave for the next generation. Will we invest in the next young woman or man peering through a microscope at next little critter, the next bit of pond scum, curious about a question we don't even know today is a question? It could be a great question that could impact all the world. And maybe you're curious about you. Now that you know how to protect your telomeres, are you curious, what are you going to do with all those decades of brimming good health? And now that you know you can impact the telomeres of others, are you curious, how will you make a difference? And now that you know the power of curiosity to change the world, how will you make sure that the world invests in curiosity for the sake of the generations that will come after us? Thank you. Thank you.
Dean Ornish.
It's great to be here today. I want to thank all the organizers for the chance to be here today, and particularly to Kunal. We tend to think of advances in medicine as being a new drug, a new laser, something really high-tech and expensive. And we often have a hard time believing that the simple choices that we make in our lives each day can make such a powerful difference, but they do. And in our work, we've been using very high-tech, expensive, state-of-the-art measures to prove the power of these very simple and low-tech and low-cost interventions. And since this is a conference on global health, I wanted to begin with that. And many people don't know that more people are dying today in most parts of the world from heart disease, diabetes, and other chronic conditions than AIDS, tuberculosis, and malaria combined. And what's happening is it's drawing and diverting a lot of resources away from things that really do need drugs like AIDS, TB, and malaria to things that can be largely prevented and even reversed through simply changing diet and lifestyle. And what's happening is that countries are beginning to eat like us and live like us and all too often die like us. And the irony is that the diet that we found that can reverse and even prevent most of these conditions is the way that most of these countries were eating before they began to copy us. And so this is still the steep part of the curve, so intervention now can make a powerful difference. And what's personally sustainable is globally sustainable. What's good for you is good for the planet, and here's why. It's so easy to get overwhelmed with all the crises that are facing us now, the energy crisis, the global warming crisis, and the healthcare crisis. Like, what can I do as one person? But something as simple as what we choose to eat every day can make a difference. In the energy crisis, for example, 20% of the fossil fuel that we burn each day goes to make processed foods, which themselves are not so great for us. It takes 10 times more energy to eat higher on the food chain. When you're eating meat as opposed to a plant-based diet, it takes 10 times more resources to make that possible. Michael Pollan calculated that a quarter pounder with cheese takes 26 ounces of petroleum and leaves a 13-pound carbon footprint, which is equivalent to burning 7 pounds of coal. So the next time you're having a burger, imagine you're eating 7 pounds of coal in terms of its impact on the planet. So does that mean you should never eat meat? No. But even if you just have a meatless Monday, know that it's investing meaning in our actions, as Adam was saying before, is part of what makes them powerful and makes them sustainable. From a global warming crisis, many people are surprised to learn that livestock consumption accounts for more global warming than all forms of transportation combined. It's responsible for 18% of the total world greenhouse gas emissions compared to the entire global transportation system that's only responsible for 13% of that. Livestock is responsible for even more of the most toxic parts of the gases. Methane, for example, from cow farts is 23 times more toxic to the ozone layer than even carbon dioxide. And nitrous oxide is actually almost 300 times more toxic to the ozone layer than let me see if I can get this slide back up, than CO2. So these simple changes can make a powerful difference in not only how long we live, but also how well we live. From the standpoint of eating meat, again, what's good for the planet is also good for you, and what's bad for the planet is also bad for you. Eating a lot of meat, a number of studies have come out recently showing that red meat consumption increases total cardiac mortality, cancer mortality, and all-cause mortality. I like this cartoon. It's the cows going off to the slaughterhouse saying, "My only consolation is that by eating us, they're killing themselves." From a health crisis, 3/4 of the $2.8 trillion that we spend each year on healthcare costs, which are really, for the most part, sick care costs, are for chronic diseases that we can largely prevent or even reverse simply by changing diet and lifestyle. This is just one of many studies that showed in large numbers of people walking a half hour a day, not smoking, eating a reasonably healthy diet, and keeping a healthy weight prevented 93% of diabetes, 81% of heart attacks, and so on. And these are probably underestimations. It's probably even more than that. Well, how do we treat heart disease in this country? Well, we generally do it with a lot of drugs and surgery. And we spend a lot of money. We spent $60 billion on angioplasties and stents in the last year that we have data on them. And you say, well, that may be a lot of money, but think of all the lives it saves. Except that it doesn't. The latest randomized trials, a total of 8 of them, were reviewed recently in the archives of internal medicine. They found that unless you're in the middle of having a heart attack, which the vast majority of people who get angioplasties and stents are not, they don't prolong life, they don't prevent heart attacks, they don't even reduce angina. And so the same is true for bypass surgery. Unless you're the 1 or 2% of people who have the most severe disease, they don't prolong life or prevent heart attacks either. That's $100 billion for 2 operations that are dangerous, invasive, expensive, and largely ineffective. Now, the cartoon says, I can operate or you can go on a strict diet. He says, well, you better operate because my insurance doesn't cover a strict diet. And that's been the problem, that with all this talk about evidence-based medicine, reimbursement is really a much more powerful determinant of how we practice medicine. Now, you find the same patterns with prostate cancer. The New England Journal of Medicine had 2 major studies, and they showed that only 1 out of 49 men who is treated for prostate cancer with surgery or radiation actually lives longer because of it. The other 48 tend to become either impotent or incontinent or both. So you take a guy who's often in the prime of life, in their 50s or 60s, find out they have early-stage prostate cancer, scare the hell out of them. They end up having an operation that doesn't really help them, but it maims them in the most personal ways. Now they're wearing diapers and can't have sex for no benefit. But the alternative is to say, well, let's just do watchful waiting. Let's just wait under a sword of Damocles for something bad to happen. And that's not very good. And so the U.S. Preventive Services Task Force recently recommended maybe we shouldn't even screen men for prostate cancer because it's too hard to know that you have it and, quote, not do anything about it. But here again, there's a third alternative. As I'll show you, when you change your diet and lifestyle, you can slow, stop, or reverse the progression of early-stage prostate cancer without having to do that. Now, diabetes is another issue. Type 2 diabetes is a global epidemic. It's a pandemic. Already, a third of Americans are diabetic or pre-diabetic. In the next 8 years, it's estimated to be half of Americans at a cost of $3.3 trillion, clearly not sustainable. Now, it turns out that lifestyle changes are actually better than drugs at preventing diabetes. This was a major study that was in the New England Journal 10 years ago, and it showed that lifestyle actually worked better than a drug to prevent diabetes. But lifestyle changes are also better than drugs at treating diabetes. This was in the New England Journal a year and a half ago. They had 2 drugs to lower blood sugar, and they found that it didn't work nearly as well to prevent the complications of diabetes as doing it through lifestyle. And the complications of diabetes are pretty awful. Heart attacks, strokes, amputations, blindness, kidney failure, and so on. But if you get someone's blood sugar down through diet and lifestyle, you can prevent all of these complications, both the human costs as well as the economic costs. And my colleagues and I at the nonprofit Preventive Medicine Research Institute have trained about 55 hospitals and clinics around the country, including 24 in West Virginia, Nebraska, and Pennsylvania. And we found that in looking at large numbers of these patients, we could get their blood sugar down to a level that we can prevent all these costs and complications simply by changing diet and lifestyle, and at a fraction of the costs, and the only side effects are good ones. So what are we doing? Well, it's what we eat, how we respond to stress, how much exercise we get just walking a half hour a day, and how much love and intimacy and social support we have. But it really goes back to a very radical concept, radical in the sense of getting to the root of something, which is what is the cause? And we spend so much time in medicine mopping up the floor around the sink that's overflowing without also turning off the faucet. And it's a simple idea, but it's a powerful one. Because if we can treat the cause, what we find, the cause, by the way, are the lifestyle choices that we make each day for the most part. And if we can make different choices, otherwise, the doctor says, take these cholesterol-lowering drugs, take these blood pressure pills, take these pills for lowering your blood sugar. And how long do I have to take them? You have to take them forever. It's like, how long do I have to keep mopping up the floor? Well, forever. Well, why don't we just turn off the faucet? Why don't we treat the underlying cause? And then when we do that, we find that our bodies have a remarkable capacity in most cases to begin healing, and much more quickly than we had once realized, because these biological mechanisms are exquisitely sensitive and highly dynamic. And it's not just lifestyle as prevention, but it's also lifestyle as treatment. Now, I began doing this work 35 years ago when I was a medical student, and I took 10 men and women with bad heart disease, put them in a hotel for a month, and we used what was then a new test called thallium to measure blood flow. And you can see around 10 o'clock there in the upper left, there's a black area where their blood flow should be going. A month later, in the same patient, in the same area, you can see there's much more blood going there. But we only had 10 patients. There was no control group. So I went back to school, finished medical school, and before starting my internship, did a second study. This time we had a randomized control group for comparison. And we found that the people who made these changes got better. The people who didn't got worse. The differences were highly significant. And we published that in the Journal of the AMA. Went to Boston, finished my medical training, moved to San Francisco in 1984, began the Lifestyle Heart Trial, and we used quantitative arteriography to measure the blockages. And you can see in the upper left where the arrow is, there's a narrowing in one of the main arteries that feeds the heart because it's clogged. Just a year later, it's wider. And because the blood flow is a fourth power function of the diameter, the actual blood to the heart was increased by 200 to 300%, which we measured using what are called cardiac PET scans. The lower left picture is the beginning. Blue and black is no blood flow. A year later in the lower right, it's orange and white is maximal blood flow. You can see these are dramatic changes. And what we found overall is that the control group, who was making more moderate changes, got worse after 1 year and even worse after 5 years. That's what usually happens. But we found instead of getting worse and worse, these patients actually showed some reversal after 1 year and even more after 5 years. This is the first time that was shown. Now, to put a human face on this, I just want to show you a 1-minute clip from a new documentary called Escape Fire that came out last month in theaters around the country. It's available on iTunes and Amazon and so on. And the filmmakers want this to be to medicine what an inconvenient truth was to global warming. So take a look.
25 years ago, I had 5 restaurants in San Francisco. It was a great life. I smoked 6 cigars a day, 10 cups of coffee, a lot of wine. It was wonderful. And I had a massive heart attack. I was in the hospital for 2 weeks. I could hardly just about walk 3 steps, and I'd have to stop and rest. I was popping 20 or 30 nitriles a day. But then Dean Ornish was starting his program to see if you can reverse heart disease through lifestyle change. And he went to my doctor and asked if he could approach me. He told Dean, how long is the program? So he said it was a year. And my doctor told him he wouldn't recommend taking me because he didn't think I would live the year. So he figured I was going to die because I was in such bad shape. And now, 25 years later, and I'm in pretty good shape.
His doctor, unfortunately, passed away in the meantime. But this is a guy who hasn't had chest pain now in 25 years, who literally couldn't walk across the street. That's why I'm so passionate about doing this work. This is the kind of thing that we see all the time in thousands and thousands of patients. So we wondered if maybe this could help prostate cancer as well. So we did a study in collaboration with Dr. Peter Carroll, who's the chair of urology here at UCSF, and the late Dr. Bill Fair, who at the time was the chair of urology at Sloan Kettering in New York. And we took men who had biopsy-proven prostate cancer, but who had elected not to be treated for reasons unrelated to the study, so that we could then divide them randomly into two groups, ask one group to make these lifestyle changes but not the other, and see what happened without being confounded by the usual chemo and radiation and surgery. And we found that the PSA levels, a marker for prostate cancer, went up or got worse in the group that didn't change, went down or got better in the group that did, and these differences were highly significant. And they were in direct proportion to the degree of change in lifestyle, which is the same we found in heart disease. The more people change, the more they improve. We looked at the effects of when we added their serum to a standard line of prostate tumor cells growing in tissue culture, the tumor growth was inhibited 70% versus 9%. And in one of the coolest slides, the more people changed their lifestyle, the more it directly inhibited the growth of prostate tumors growing in tissue culture. And through John Krahanowicz's lab here at UCSF, we did MR spectroscopy showing the tumor activity, shown in red here, was diminishing in this patient after a year, as well as the PSA going down. So taken as a whole, this is really the first and still the only randomized trial showing that the progression of men with early-stage prostate cancer can be slowed and stopped and often even reversed simply by making changes in diet and lifestyle. So we wondered what some of the mechanisms might be to help explain that, and we found that gene expression was changed in over 500 genes in just 3 months. In effect, turning on or upregulating the good genes that protect us, downregulating the bad genes that cause inflammation and oxidative stress, and also the RAS oncogenes that promote prostate, breast, and colon cancer were downregulated or turned off, hundreds of them in just 3 months. And this is what's called a heat map. You can see on the right are the oncogenes, and red is mostly turned on. 3 months later, green is mostly turned off. It's amazingly powerful. We did a study with Dr. Elizabeth Blackburn, who won the Nobel Prize for discovering telomerase, which is an enzyme that repairs and lengthens damaged telomeres, the ends of our chromosomes that control how long we live, and also Dr. Lissa Epel, who'll be presenting later today. And what we found was that the telomerase could increase by 30% in just 3 months, and no study has shown that. And we're about to publish the 5-year follow-up and show that the telomeres themselves actually get longer when you make these changes. And this will be the first study to show that as well. If this was a new drug that could lengthen your telomeres, it'd be a multi-billion drug, billion-dollar drug overnight. But it's the same lifestyle changes that do all these things. It's not like there's one for diabetes, another one for heart disease, and so on. And we found, again, the more lifestyle changes people made, the longer their telomeres got. We also found that angiogenesis changes. This is the first study to show that. We found that we could downregulate VEGF, which tumors secrete to cause blood vessels to grow and feed them. Drugs like Avastin and Nexavar inhibit VEGF, but it costs $100,000 a year per person to take. This is, again, for free. It's just the same lifestyle changes. So the more we look, the more mechanisms we can invoke to explain why these changes are so powerful and why they make such a great difference. So our genes are our predisposition, but our genes are not our fate. I find that very empowering. We also found that this was not only medically effective, but also cost-effective. Mutual of Omaha found they saved $30,000 per patient in the first year. Highmark Blue Cross Blue Shield found they could cut their costs by 50% in the first year and by an additional 20 to 30% in years 2 and 3. And finally, a year and a half ago, after 16 years of review, Medicare agreed to cover our program, which was a real game changer because, again, reimbursement is such a primary determinant of medical practice. If you change reimbursement, you change medical practice and even medical education. So we're in the process now of training hospitals and clinics and sites around the country. If you're interested in learning more about our work, go to our website, or if you're interested in being trained in this. And our goal is to we train a team of 6 people, a doctor, a nurse, a stress management specialist, essentially a yoga and meditation teacher, an exercise physiologist, a dietitian, and a psychologist to work together as a team with the doctor as quarterback. And it enables us to reclaim our role as healers and not simply as technicians. Vinod Khosla, who'll be speaking later today, was quoted in a very controversial statement that, we doctors are going to get replaced by an iPhone app before too long. And if all we are just a collection of algorithms, then that's true. But we're more than that, and it allows us to get re-enchanted with medicine and to reclaim our role as healers and not just technicians. We train the St. Vincent de Paul Homeless Shelter here in San Francisco. There are over 20,000 patients who've now gone through our clinic in the last year and a half there. And now with the Medicare reimbursement, it's financially stable, and we can now clone this around the country without needing ongoing philanthropy to support that. But the other big epidemic, besides heart disease and obesity and diabetes, is depression and loneliness. And study after study have shown that people who feel lonely and depressed are many times more likely to get sick and die prematurely than those who have a sense of love and connection and community. We found we could cut depression scores in half simply by changing diet and lifestyle. comparable or even better than what you get with antidepressants. And you know, because there's been a breakdown in the social networks that used to give us a sense of connection and community. Many people don't have a job that feels secure, a neighborhood where you have several generations of neighbors living together, an extended family, or even a nuclear family that you see regularly. And we all know that these things affect the quality of our lives, but they actually affect our survival. And you know, Nick Christakis' work found that these social networks are so powerful that if your friends are obese, you're 45% more likely to be obese. If your friends' friends are obese, 25%. If your friends' friends are obese, even 10%, even if you've never met them. And you can see this with other behaviors as well. That's how powerful these are. So the last thing that I want to say is that anything that creates a sense of trust leads to intimacy, leads to healing and meaning. Even the word healing comes from the root to make whole, yoga, to yoke, to unite, to bring together. These are old ideas that we're rediscovering. And you find this as part of all spiritual traditions: altruism, compassion, forgiveness, and love, not to get some external reward in the next lifetime, but that's what frees us from our isolation and our depression and our suffering here and now. My dad died a few months ago. My mom had a stroke, a debilitating stroke shortly thereafter. My dog of 14 years got bit on the nose by a rattlesnake. This is on the last few months, and died. You know, life is short and life is precious. And what I'm most interested in is not simply unclogging arteries or showing all these kinds of things, as interesting as they are, because we're all going to die. The mortality rate is still, 100%. It's one per person. I got profoundly and suicidally depressed when I was in college. That was my doorway into this area. For someone else, it might be a heart attack or a stroke. But change is hard, as Adam mentioned earlier. But when you're in enough pain, suddenly the idea of change becomes more interesting. And what I'm most passionate about and what I find most interesting is how we can use the experience of suffering as a doorway for transforming our lives and finding meaning. And then we can often get curing, but we can always get healing. Thank you so much.
So let me talk about really coming down to the simplistic idea of slow and fast aging. So age is the biggest predictor of when we die, period. But within that, tremendous differences, because underlying age is biological age. Biological age is very elastic, as we know. So what are the drivers of biological age? Some people age in dog years much faster than others. And we now know some of these mechanisms. Telomeres is just one mechanism. There's no one cellular aging mechanism that is the driver of aging. They work together. They're all super tightly correlated. Telomere length, which I'll talk about, is best friends with mitochondria, reactive oxygen species, other aging mechanisms. But we can measure telomeres very easily. We now know that telomere length is causally predictive of disease. So it's not just a correlate. It's not just something that gets worse with age. It's genetically, people who have genetically short telomeres tend to have much higher odds of early onset cardiovascular disease, dementia, other diseases. So what is a telomere? So there is our chromosomes. In those strands are our genes that make us who we are, that create every protein that is our body. We must protect those genes. Aging is about DNA damage. It's damaging things so that we're creating worse proteins that can't repair, et cetera, as we age. So how do we protect the genome? One of the major focuses is this cap at the ends of our chromosomes, the telomeres. It's just a wound up strand of base pairs that is extremely sensitive to our environment, our biochemical environment in the cell, as well as you'll see our environment outside of the body. It is a canary in the coal mine. It is reading out, it is interpreting signals from our mind, from our behavior, from our environment, because it's a survival mechanism. When we're under stress, the enzyme telomerase that protects telomeres increases and protects the telomere. As we age, telomeres shorten. They can't always replicate as cells divide. And so we need to keep our telomerase enzyme active and healthy throughout our lifespan. So telomeres shorten slowly, but what's fascinating about them is they're not something that we just think about after 50 as we get sick. We inherit telomere length both genetically but also environmentally. It's directly epigenetically transmitted from our parents, meaning it is shaped by our parents' life experiences as well. So let's talk about the big drivers. Most of our behavior in life is influenced by these two big forces, love and social connection, fear, anxiety, and feeling unsafe. So these are big drivers, and it turns out that telomere length is also associated with these two big forces. So let me get a little bit more specific. What maintains telomere length? Childhood. Why am I mentioning childhood? We've had it, it's done, we can't change it. But childhood is pregnancy and childhood are critical periods that shape our telomeres and rate of cell aging for life. So I'm telling you this for two reasons. One is many of you may be testing your telomeres, which you might find out you're short and you're doing everything perfectly. How could that be? Well, early childhood adversity and especially pregnancy in the womb leave scars on telomeres. We're shorter for life. That's okay. It doesn't matter where you start. It matters what you do now and every day because we can maintain our telomeres over time. So I'm also telling you this because whatever you do, your intervention, your program, your spa retreat, pregnancy is a huge window into public health, into the future. Pregnant women are the gateway of health, and of course, women of childbearing age. And so they're very motivated. This is a tremendously important market to think about because boosting women's health during pregnancy is going to have tremendous compounding effects on both her health as well as her offspring. So habitual pathways in adulthood. So we shape our behaviors and our stress responses largely through our life experiences throughout growing up. And so here we are as adults. We have canalized reactive pathways, or maybe we have more resilient and calmer stress pathways. Regardless, these are important to think about because how we respond to stress every day adds up over time. And it's these daily habits that I want to talk about. So one thing we've studied is these psychological states. You guys are all experts in the behaviors, the behavior change. I'm not going to tell you anything too new about what to do in terms of what telomeres like for lifestyle. Because you already are probably doing them, and you know what they are. And they're the same things. What's good for the heart is good for cell aging, for the telomere. But it's the mindset that's probably new to most people. So threat reactivity, when we feel unsafe, we have bigger, more exaggerated stress responses that recover slower. And in contrast, we can have positive stress responses, and we can there are mind tricks to shape our stress responses so that we're having more of a challenge or positive response. Example, lion chasing gazelle, both having huge stress responses, very different cardiovascularly and in terms of how high the cortisol is. So that lion is having a positive stress response. He's going to gain something. Has tremendously efficient cardiac output. Gazelle is completely vasoconstricted, ready to be bitten, ready to vasoconstrict so he doesn't bleed to death, threat response. So that might be relevant to giving a speech, or when we do acute stressful things, we can try to shape our acute stressful response. In real life, we are constantly coping with chronic situations. So it's a different type of stress as well that we need to think about. And that is thinking about how resilient we are to adapting to things we cannot control. And so we think about stress being a source of something that strengthens us when we have appropriate resources. What doesn't kill us can make us stronger. So that's positive stress. We've also been studying mindset. So when where's your mind right now? This second. Yes, thank you. And don't tell me if you don't need to tell me what other things you're thinking about. So when we are completely engaged and focused, this is obviously a positive state. Our physiology is responding. This is a state of mindfulness that's associated with wellness. So I'm talking about informal mindfulness in the moment. Mind wandering is very common. We mind wander half of the time. What I want to point out to you is negative mind wandering. So notice if you're having thoughts of, I don't want to be where I am right now. I don't want to be doing what I'm doing right now. I wish things were different. That is unhappiness. That is suffering. And that is a state that we can notice by becoming aware of our thoughts and be changing. So in our observational studies, negative mind wandering is associated with shorter telomere length. And in our mindfulness interventions, of course, that's one of our targets. We're trying to improve engagement and presence in the moment. Presence is associated with longer telomeres. So now we get to the health behaviors. What's damaging? What's protective? So we want to think, the guiding framework is this. Telomeres are damaged by oxidative stress, inflammation, stress hormones, high levels of insulin resistance. They're protected by restorative behaviors, sleep, high vagal tone, so when our nervous system is on restorative mode, and anabolic growth factors, but not unnatural ones. So just to show you visually, how can we slow aging? Most of these studies I'm showing you are cross-sectional, but there's an emerging body of intervention studies as well that show us that the telomere maintenance system is flexible, it is malleable. We can boost our health in the short term. Sedentariness is related to shorter telomeres. Exercise interventions appear to boost telomere health. Sleep quality. I don't know who last night did DeStefano's sleep ritual, but I slept better, thank you. Sleep quality is critically important. So people who get, well, duration's important. People who get less than 7 hours in the night tend to have shorter telomeres, but the quality of our sleep is also important. So putting ourselves into a bedtime state, having a sleep ritual that actually shifts us from the active doing mode to a more peaceful, ready to restore mode is critical. So I loved the sleep ritual. I thought that was helpful. Rituals are completely personal, so it's what works for you. Eat your vegetables. Antioxidant diet. It's so simple, but it is so correlated with longevity. It's in all of the 100 of diet studies, it's about vegetables, fruits, and antioxidant diet, a whole food diet. Meat, sugars, soda, these all shorten telomeres. Extended mind, my favorite new concept. I must share with you what extended mind is because you all know this and live this, and it's a relatively new social cognitive neuroscience concept, and it's this. Our thoughts are not just in our head. Our mind is outside of our body, and it is shaped by social interactions that we're having with someone else. It is shaped by the environment. That's how we think. Our thinking is emerging in moment to moment based on our surroundings. And so what does that mean? That means that we're influenced by people around us who are very stressed, but the converse too, we can use this for good. When we are emotionally contagious to each other, and there's a tremendous body of research showing how our physiology as well as our felt emotions are correlated with each other and move together, and we influence in each other in these implicit, unconscious ways that are so important. So being well together is really the only solution here. It's not about individuals. We think we're separate and independent. That's a complete false perception. So the interventions you do, you create these settings that are so powerful. People walk into a retreat center, and things are changing immediately. And it's that conditioning to these environments that is so restorative that we want to share with so many people who don't get to have these safe havens and these experiences. So think of daily restoration. Is it the daily grind? Are you having these repeated stress reactions every day that are wearing away base pairs? Or are you building in restoration? And these healthy behaviors, they're small, they're tiny, but they add up over years. So last topic, can meditation increase telomere health? Of course, this is one of our favorite topics, and we can learn from our pets about being mindful. So there's now a body of studies showing that different mind-body activities, there's absolutely nothing unique about meditation studies. We find this with Qigong and different types of yogic meditation, et cetera. And common ingredients, focused attention, calm, sympathetic arousal, et cetera. So these appear to boost the enzyme telomerase, and in more long-term studies may also stabilize or lengthen telomeres. We have a paper under review that shows 3 weeks at a meditation residential retreat actually lengthened telomeres. So that's probably the shortest I would ever go, and that means they're eating healthy food, there's no computer, so that's a pretty intense intervention, the shortest I would look. I'm going to tell you briefly about one of the funnest studies I did where Deepak Chopra opened up his retreat center, Study Us. I think this is transformative. I've been doing this for 10 years, and people tell me it changes their lives. So we went in and we measured well-being, we measured blood before and after the week, and the beauty about the study is that we got to randomize people to staying at the pool or the resort, La Costa, or being in a meditation retreat for a week and learning a new skill. They were complete novices. So what do you think we found at the end of the week in terms of well-being, vitality, mindfulness, decreases in stress? So we had the 2 groups that randomized. We also had a group that was experienced meditators, signed up, they were there anyway. We, asked them to be in our study, give us a little blood, fill out these questionnaires, help signs. So at the end of the week, we found dramatic increases in well-being, 60% improvement on all the well-being scales, and reductions in stress and depressive symptoms. And this was really exciting. We don't see changes like this in other types of interventions. This is big, and it happened in all 3 groups. So what we're looking at is a vacation effect. People unplugged from their life. They came to this resort. Didn't matter what they did. They didn't have computers. They were living a more restorative daily life. And then we looked at what was happening in the blood. And in gene expression, we found dramatic increases in the expression of dramatic reductions in the expression of genes related to stress and immune function. So basically, all of that defense response that we've got on all day to fight with all these demons and tigers, that was just dampened completely in all 3 groups. So that was the huge finding. We call that the vacation effect. And then the other finding was the telomerase did boost, but only in 1 group, and it was the experienced meditators. And that is a pattern in meditation research, that novices show some improvement, but if you're looking at brain waves, if you're looking at gene expression, it's people with experience that then are studied acutely have more dramatic responses. So it's just exciting. I know Mark Cohn, sitting in the front, is one of the few people who is actually doing a lot of these retreat studies. And it's a wonderful thing to partner with scientists. I know my colleagues would jump at the chance of studying one of these intense interventions. Retreats are an extremely powerful catalyst for change, and we need more research to show this. I've just learned so much being here, and of course my first question every time I hear about one of hydrotherapy or one of these interventions is, where's the data? We really need to be studying these amazing retreats. My last point is thinking about when you think about how you want to live your day, which is what matters, right? We are all going to die, and we don't know when. We can only hope for a good health span. We can't control our lifespan. We can control how our health is and hope that we can stretch it to the end. So thinking about the end game sometimes helps us change our behavior in the moment. So let me just ask you, not to be morbid, to think about what you want written on your epitaph, on your tombstone. What is your epitaph? None of you are probably thinking, I wish I spent more hours at the office. I wish I made more money. No, at the end of life, it's about love and relationships and connection. Have you loved fully? Have you lived fully? Have you let go of the small things? And those are actually the ingredients for our daily life as well, that shape our behaviors towards slow aging, towards an expanded health span. So we can't forget the health behaviors, right? They're necessary but not sufficient. Eat your vegetables. Telling people that doesn't help because, as we've been hearing over the week, a theme is people do well when they can. When they can. When their mind and their environment allows them. So present mindset appears to be related to slower telomere attrition. So in a way, it's making time more elastic. even though seconds are equal. Biological aging responds to our mindset. Being present allows us to connect with other people. So that leads us to this extended mind idea. Wellness within can allow wellness between us, between people, and wellness together, neighborhoods, communities. And of course, meaningful work and purpose in life. Purpose in life, boosts in purpose in life after interventions is associated with boosts in telomerase. So our cells are listening. They know what matters most. Thank you so much for listening.